my name is David, and today I'll be going over the ACT science test for Hamilton Test 106. Let's get started. Passage 1, Introduction. This science passage looks like a data representation passage because it has a short blurb right here, and then it has two figures. So my plan of attack for this passage is to go straight to the questions and then try to answer them as fast as I can. And then if I need to, read back over the introduction. All right, let's get started. Question 1. According to figure 1, the majority of the nearby stars are which of the following? Less bright, more bright, the same brightness, or vary greatly in brightness throughout the year. Okay, so for this one, figure 1, I notice it says HR diagram, 50 nearby stars. We have luminosity here, which is brightness, and then surface temperature, which is K. And I also notice that there's these diagonal axes that are RS. So as of right now, if you did not know what luminosity was, you would have to read the passage. So in the passage, it says astronomers classify stars according to luminosity or brightness. Okay, so then it's brightness, and we're talking about brightness for figure 1, so I should be looking at luminosity. Now, you may not know what the brightness of the sun is, so if you keep reading, it says luminosity is expressed in solar units, where 1 solar unit is defined as the luminosity of the sun. So 10 to 0 would be the same as 1. So that means anywhere along this line would be as bright as the sun. If you look though, the majority of the dots are below that line. So that means that most of the stars are going to be less bright than the sun, which is why the answer should be A. Question number 2. Blue giants. Whenever you see an italicized word like that, that means it's a new term or something that you will based on the information provided in the question. But blue giants are bright stars with a luminosity. Okay, we just saw that for question 1, approximately 10,000 times that of the sun. So looking back over here, 10,000 would be 10 to the 4th. And then if you keep reading, the surface temperature of approximately 20,000. We saw before that the x-axis was 20,000. So I kind of feel like we're looking at this point right here. All right, according to figure 2, the blue giants have a radius of what? Okay, well, it says figure 2, not figure 1. So then we're going to move over to figure 2. And we're going to take the same point, which is right about here. And what is that between? Well, we're looking for the radius, and the radius was not the x-axis or the y-axis, it was these diagonal axes. So looking at these axes, I can see that it's between 100r and 10r, where r is the radius of the sun. So let's look if we have an answer choice that matches that. F, close to the sun's radius, so that'd be about 1, so it's definitely more than that, because that'd be along this line. Less than 10 times the sun's radius, so that'd be below this dotted line, which is it's above. Between 10 and 100 times the sun radius. That's exactly what we're looking for. So the answer is H. Question number 3. White dwarfs, so same thing as before, question 2, it's italicized, are small stars between 10 to the negative 2 and 10 to the negative 4 times as bright. So this is again referring to the y-axis. With the surface temperature between 10,000 and 40,000, it's referring to the x-axis. If the data in figure 1, so we're going to be looking at figure 1 here, is representative of all stars in the galaxy, approximately what percent of the stars in the galaxy are white dwarfs? Okay, so between 10 to the negative 2 and 10 to the negative 4, and we're going between 10,000 and 40,000. So we're looking at these number of stars, which is 1, 2, 3, 4, 5, 6, 7, maybe 8. So we have 7 or 8 stars out of 50. I know it's out of 50 because it said there are 50 nearby stars, so you didn't have to count up all the dots. Now, if you couldn't divide 7 or 50 quickly, one thing you could do is times the bottom and the top by 2, and that makes the bottom out of 100. So then I get 14 or 16%, which looking at the answer choices, 15 is right in the middle of them, so that's pretty close to what we want. 10 is too small, that would only be 5 stars, and 20% would be too big, that'd be 10 stars. So that's why the answer was B. Question number 4. According to figures 1 and 2, so we're going to have to combine these two graphs, as the size of the radius increases, so as R goes up, the surface temperature does what? So you have to remember that the radius increasing is going along this direction. So let's see if we see it going up or down or a mixture of the two. So looking at along this, as the rays goes from here to here, it does seem like the temperature went from 2,500 to 5,000. Does that kind of make sense? So it seems like the temperature does go up. However, as you continue going up, it sometimes seems that the temperature can decrease. So for example, here when the rays was very small, we have some future temperatures that are cooler. Over at figure 2, we have again, as the rays goes up, the temperature seems to be increasing, but then way over here, we have a group that are very large and cool. So I would say that the best answer here is increase and decrease. Alternative answers that I might pick would be that there's no clear trend because it does seem to go hot, then cold, then hot again. But the best case is increasing and then decreasing. Question number 5. Compared to the nearby stars, the brightest stars in the night sky are which of the following? So we are comparing the nearby stars to the brightest stars. So looking at this chart, I could look at a single data point, but it might be better to look at the nearby stars as a whole. So I'd say that the most nearby stars are kind of around 1 to less than 0.1 R and around 5 to 10,000 in temperature. Whereas if you look at figure 2, majority of them are right here. And again, there's partial over there, but most of them do seem to be above 1 radius, maybe like a little bit greater than that. And the temperature seems to be between 10 and 20,000 for majority of them. So looking at that, I would say that majority of them are definitely larger. So it's not A or C. And then is it cooler and hotter? Well, if you compare a single data point, you might argue that it's cooler. But then there are some points that are both hotter and larger than this data point. So I think the best choice is to compare them as a group or as a whole. So that is why I'd pick D, that most of these stars are both hotter and larger than the ones that are nearby. Question number 6. According to the figures, a star with a surface temperature of 6,200 Kelvin and a luminosity of 10 solar units should have a radius that is approximately what? So they both will have the same surface temperature and luminosity, so you could use either chart here. Whereas if it did say something about the bright star and nearby stars, you would want to pick one of them. So let's look at where 10 luminosity is. So it's along this line. And then 6,250. So 7,500 is right in the middle. So 6,000 would be a little bit closer to the 5,000. And that should be right around there. So let's look at the different answer choices. Well, we have slightly larger than that of the sun. Well, it is larger than 1 radius of the sun, so that seems reasonable. G, slightly less than that. No, it's definitely bigger. 10 times larger than that of the sun. Well, I would say that that could maybe seem reasonable, but that might be if it was 5,000 Kelvin, would be 10 times. So I don't think that's quite what we want. And then less than 1/10, that's going in the opposite direction. So that makes F the best answer choices, where it's between 1 times radius and 10 times the radius. Introduction to passage 2. So we're now starting passage 2. This is about garlic, and we're testing its effectiveness on bacterial growth. Looking at it, there's some text here and a single graphic. So it suggests it's a data presentation passage. So once again, we're going to go straight to the questions. Let's get started. Question number 7. During the 7-day period, the rate of change, so it's probably talking about slope, in the diameter of the E. coli colonies was greatest for which of the following dishes? So we're looking for the rate of change over 7 days. So for dish A, it's 7.1 over 7. So that's about 1. B is a lot bigger. It's 50.1 over 7, which is about 7. And then dish C didn't grow at all, so that had no change. So the one that had the greatest rate of change would be answer choice B. Question number 8. Based on the information in the passage, which would be the most likely outcome had the garlic been roasted before the juice was extracted? Okay, so we'd had through the passage to find information about that. It can be helpful to read the answer choices first, though, to maybe clue you what you're looking for. So F says the E. coli growth would have increased twice as fast because the heat would have sped up, so I might be looking for heat. The dish C would have developed a different strain of bacteria. Based on my understanding of biology, that seems unlikely because roasting garlic doesn't make a new species of bacteria come about, but we'll look for that. C, the amount of bacteria growth in C would be similar to the dish B because the allicin would have been inactivated. Well, that does seem kind of likely based on my knowledge in biology. Usually if you apply heat to a protein, it denatures it or deactivates it. And then J, there'd be no perceived difference in the amount of bacteria. Okay, let's go be about it. Garlic is used in culinary dishes. Cool. But again, that's not about what we're talking about. Low is vitamins, minerals. All right. Contains allicin. Oh, an antibacterial sulfur compound that's released in garlic is chopped or crushed. In order for allicin to be affected, it has to be consumed raw, not cooked. So that's kind of telling me that if we cook it, we'll probably break the antibacterial sulfur compound and make it not work. So I feel like the answer should probably be H, but we can confirm it by reading further. To set up the experiment, they had 3 separate test tubes. The first tube A contains pasteurized milk. So pasteurized milk is usually they've boiled it or heated up so that way it kills all the bacteria or most of the bacteria. Question number 9. What is the most likely explanation why E. coli bacteria cells developed in Petri dish A? Well, because we had to read the introduction based on question 8, I kind of have a good guess on when that might be, and that would be that there's E. coli getting added by the surroundings. Let's see if we have an answer choice that matches that. Some of the bacteria from the environment contain features A, most likely during the incubation period. That sounds pretty promising. The fat in a whole milk most likely contained trace amounts of E. coli that grew there throughout the course of the experiment. I don't think that's true. The allicin used in dish A was defective. We didn't use allicin in dish A. The lack of sunlight where the peaches were grown stored stimulated the bacteria growth. They didn't say it grew it in the darkness or in light, so we can't conclude that either. So the most likely scenario is that it got added from the environment. Question number 10. The table indicates the greatest percent increase in the diameter of E. coli in dish B occurred between which of the following time periods? So we have 3 to 4. So it grew about 3.6 centimeters out of 6.6. So that looks like about 50% increased. Day 4 to 5, it grew by, that's about 13 out of 10. Well, that's definitely more than 50%. It's like probably like 130%. So F is out. Day 5 to 6, that grew by about 16 or so. Well, that's out of 23, so that's less than G. And then 6 to 7, it grew by about 11 or so, 39.8. So that is a lot less than G. So that's why G was the correct answer. You may also notice that I didn't use exact numbers or I didn't find the exact percents because I don't have a calculator. So being able to approximate or estimate the values is very helpful for doing the math on the ACT science. Question number 11. Based on the table, which of the following graphs best demonstrates the bacteria growth in each of the dishes over the experiment? Cool, so we're taking this table and we're making it into a picture. Well, I know that dish B grew the most, so I would look for the answer where it had B be the highest. So for answer choice A, dish A is the highest, so we can eliminate A. In this case, dish B is the highest, so that's a good sign. In this case, dish B is the middle one, so it's not correct. And then this one, dish B, is probably near the end, but also in the middle. So we wanted answer choice B because it would have dish B would be the greatest growth, dish C the least growth, and dish A in the middle. Question number 12. A student predicted that the dish containing both the E. coli specimen and the garlic juice sample would initially develop a bacteria colony, but that the allicin in the garlic juice would eventually kill the colony by the end of the week. Do the results in table 1 support the claim? Okay, so this is code to say look at dish C because that was the only one that had allicin and the bacteria at the beginning. So let's look at dish C. In day 1, it didn't have any growth. In day 2, it didn't have any growth. So it kind of looks like to me that the allicin killed the bacteria colonies right in the beginning. So let's look at F. Yes, because there's no bacteria colonies on day 7 dish. So these are called half-truths, where the last half is a true statement, but the first part is not true. There was no growth of bacteria to begin with, and then having it killed, it was never there to be, it never grew to begin with. So that means we can cross off G as well because it has the yes in there. And again, you can see it did not grow exponentially. So H, no, the garlic juice appears to prevent E. coli from growing at all. That sounds pretty good. We have the no and the fact that it did not grow at all and then die. And also it appears to have prevented it. And H, no, the garlic juice has no significant effects on the development. Well, it definitely did because there were no bacterial growth or colonies in here. So number 12 is kind of tricky. If you fell for F, that's because the second half of it sounds pretty true, but you need to make sure to consider both parts, the beginning and the end. And that does it for passage 2. Intro to passage 3. My first instincts for passage 3 is that this is probably a research passage, also known as a research summary passage, because it has multiple experiments. Looking at it has pulleys, so this is physics, but as we've talked about before, usually the subject matter does not really affect us. All right, let's get started. Question number 13. Suppose experiment 2 had repeated using the 3-pulley apparatus and used a 16 Newton weight. Which of the following values would be closest to the expected force measured by the spring scale? All right, so I look at experiment 2, I see the 3 pulleys, and I see a bunch of numbers, but I don't know what A, B, C, D is. If you look at table 1, though, it shows you that A is 1, B is 2, C is 4, D is 8. So since we're looking for a 16 Newton weight, that means we can pretend there's an E there and double that. All right, well, let's see if we can find a mathematical pattern. Well, I know that it should be greater than 2.67, but they're all greater than 2.667. So let's see if there's some mathematical relationship. When C was 4, it was 1.33. When we doubled it to 8, it went to 2.667. So I think we can assume that's going to follow the same trend. So if we go from 8 to 16, we doubled the weight, which means we should double 2.667. And that's greater than 5.0, which makes it answer choice E. Question number 14. The mechanical advantage of a machine is defined as the ratio of which the machine multiplies the force applied by the user. According to tables 1 and 2, how is the mechanical advantage of the block and tackle pulley system related to the number of pulleys used? So for experiment 1, the student had to use a force of 1 Newton to lift A. But in experiment 2, for the same weight A, when there were 2 pulleys, it did half a Newton. When there were 3 pulleys, it was a third of a Newton. And when there were 4 pulleys, it was a quarter of a Newton. So it seems like to me, the number of pulleys is related to the mechanical advantage. So let's see if we have an answer that matches that. The mechanical advantage is 4 times the number of pulleys. Well, no, it wasn't 4 times 2, and then an eighth of the force. The mechanical advantage is double the number of pulleys. Well, 2 times 2 would be 4, so then 1 divided by 4 would be 0.25, not 0.5. The mechanical advantage is used the same number of the pulleys used. All right, that sounds pretty reasonable. 1 divided by 2 is half, 1 divided by 3 is 0.33, 1 divided by 4 is 0.25. And this is true for any of the weights you use. So that's why the answer was H for number 14. Question number 15. Suppose a block and tackle pulley system had been assembled using 5 pulleys. The amount of force required to support weight C used in the system is closest to which of the following? Well, as we just discussed in question 14, we can use the mechanical advantage. to figure this out. So we assume that mechanical advantage is 5. So 5 is equal to the force output divided by the force input. Because it's weight C, we need to have at least 4 newtons. And so then we can solve for the force going in. All right, which should be taking the weight and dividing it by the number of pulleys, as we talked about with answer choice 14. So let's see, which of the following is 4 divided by 5? Well, that's less than 1, and there's only one answer less than 1. Or you could do times both the top and bottom by 20 to make it 80 out of 100, which is 0.8. And that is why for 15, the answer was. Question number 16. The work performed on an object is defined as the product of the force applied to the object and the distance the object traveled. So then work is equal to force times distance. According to tables 1, 2, and 3, the work performed in experiment 3 on weight B. So we have weight B right here. All right, when it was weight B, that was a force of 2 newtons. Let's write that down. All right, when we have number of pulleys, so it would be 2 times 10, which is 20. When we use 2 pulleys, the force required is 1 newton. So then there'll be 1 newton times 20, which is 20. Well, what about for 3 pulleys? For 3 pulleys, it'd be 0.666 times 30. I'm guessing that's also close to 20. So it seems like the work is constant. Say the same as the number of pulleys increased. That seems right. Increase as pulleys increase. No, it stayed the same. Decrease. No, it stayed the same. Increase and decrease. No, it stayed the same. So that's why for number 16, the answer is F. Question number 17. The main purpose of experiment 2 was to determine which of the following. So again, you can read the introduction here, but we already kind of see that we had different number of pulleys and we saw different questions that seemed to be about the mechanical advantage. So that's what I'm going to be looking for. The force required to support each weight with no mechanical advantage. Well, no, that was actually experiment 1. The mechanical advantage conferred by varying the number of pulleys. We varied the number of pulleys. We saw the different change in force. So that sounds pretty good. But we can check C and D just to make sure. The rotational inertia effect. Well, we didn't talk about rotational inertia here, so it's not C. And then by varying the length of the rope. No, that was actually experiment 3. So we can cross off D as well. All right, for 17, the answer is B. All right, question number 18. A student hypothesized that a block and tackling pulley system with 8 pulleys would require 2 newton force input to support a 16 newton weight output. Does the data reporting experiment 1 and 2 support this student's claim? Well, as we talked about earlier, the mechanical advantage will be the force out over the force in. So let's see if it's true. So we got 8 pulleys, which is equal to mechanical advantage. So 8 is equal to force out is 16 over the force in, which is 2. And that's true. So we want yes. So we can eliminate the no's. Yes, because the force required was directly proportional to the number of pulleys. Force required was not directly proportional. Actually, the force went down as we increased the number of pulleys. So it went from 0.5 to 0.3 to 0.25. So the force needed decreased. So inversely proportional. Yep, that's perfect. So as the number of pulleys increased, the force decreased. And that's why the answer is G. And that's it for this passage. Congratulations. Intro to passage 4. This is another Reese's passage, and we're going to be looking at it because it's an experiment 1, 2, and 3. We got different charts, tables, and graphics. So we're going to be talking about BD and how does it affect a specific type of tree frogs. But on that, though, we're going to get started with the questions. Let's go. Question number 19. Which of the following variables was kept constant in experiment 1? So we're going to be looking at table 1 here under experiment 1. All right, the number of times we're exposed to BD, I can't tell from the table. The temperature, I can't tell. The amount of BD that they're exposed to, I can't tell. But the length of time, well, I can see for A, B, and C, they're all monitored for 6 weeks. So my answer is probably D. However, if we want to verify it, we could read the description of experiment 1. So they added the kitchen by Moses into 3 groups of 100 tree frogs by swabbing the undersides with 1 ml standard solution containing BD. Group A were monitored for 6 weeks, an environment kept at a constant 20 degrees temperature. Group B was 30 degrees Celsius, so we did not keep the temperature the same. For 2 weeks, they were then re-exposed with the BD, so then we did not keep the amount of BD they're exposed to the same, and the number of times. So that quickly limits A, B, and C and leaves us with answer choice D. Question number 20. According to the data from experiment 3, has the number of exposures to BD increased? Well, we were just reading about that. So group A had it 1 time, B had it 2 times, and C probably had it 3 times, but we can check. Group C were placed in 30 for 2 weeks, then re-exposed to the solution. They were placed back in 30 for 2 more weeks, and then re-exposed a second time. So that is a total of 3 times. What happened to the number of leukocytes per gram of body weight in the frogs at 6 weeks? Well, looking over the process of 6 weeks, group C was the dark one, kind of goes like this. Group B kind of goes up. So, but B, which was only exposed 2 times, did not increase as much as group C did. And let's see what group A did. Group A went downward. All right, so definitely it looks like the group with the most exposure to BD had the greatest increase in leukocytes. So the answer should be F. Question number 21. If a frog has been exposed to BD a 4th time and then placed into a test tube describing experiment 2, what percent of the time would it most likely be found on the sterilized side? All right, so we saw that for question 20, we had A was 1, B was 2, C is 3. And looking at experiment 2, I can see it goes A, B, C like this. So I would imagine that D would be somewhere along here, which is probably below 20% or 25% on the BD side. However, it didn't ask for the BD side, so 20% is probably the BD answer. We're looking for the sterilized side, which would be the other side of the dish. So 100% minus 20% would give us 80%, which is why the answer was D instead of A. All right, question number 22. According to experiment 3, what was the average number of leukocytes per gram body weight found after 6 weeks in frogs that have been exposed to BD twice? Well, that means we're looking at group B. All right, group B, which is light one at 6 weeks, which is roughly around here. So I'm going to kind of draw it over. So it looks like a little bit above 500. So not 100, not 250, 500, it's not quite right, but 750 is way too much. 750 would be kind of like the group C at 5 weeks. So the best answer here is H. Question number 23. Which was found as a possible weakness in the design of experiment 2? So weaknesses in experiments are usually defined as not controlling for some variable, or maybe looking at a smaller group, or looking at a subset. So let's see which one of these best matches it. The amount of time that each frog spent in the chamber was different. No, it said the surviving frogs from experiment 1 were placed in the compartment test chamber for 24 hours, and that's true for all of them. So that's not quite a weakness. The number of frogs used from groups A, B, and C was different. Well, it doesn't say the number of frogs here, it just says the surviving frogs. And one thing you might then look back at in terms of table 1 is you may remember that for group A, only 10 of them survived. So they're only having 10 of the frogs in here. Whereas group C, they had 72 frogs in here. So the extra frogs may have made it crowded, or maybe they're more immune to it, so they're okay with going on either side of it. All right, so that definitely seems like a weakness. The BD positive is larger. Nope, the right half was coated with the substrate contained BD, and the left half was sterile, so that's not true. And the frogs use visual cues to distinguish BD positive in sterilized floor areas. It doesn't mention visual cues here, but I mean that hypothetically could be a situation. I think, though, the more likely answer is that the number of frogs used in groups A, B, C was different, because we're comparing 72 frogs for group C to 10 frogs in group A. So I picked answer choice B for question 23. Question number 24. The data strongly indicates that the number of leukocytes per gram body weight in the frogs increased the most rapidly after how many exposures to BD? So because it's leukocytes per gram body weight, I know I'm looking at experiment 3. Let's compare that. Well, weeks after initial exposure of BD. Well, before or 0 exposures, there's probably around 250 or less or something like that. We don't really know. So I'm not going to pick F because I don't quite know what it should be. But after 1 exposure to BD, well, it starts here. It kind of looks like some go down. So that might be the initial reaction. They might be dying from it. Let's look at 2 exposures. So the second exposure occurred after 2 weeks. So then looking at the 2-week or the second week marker, it does seem like they start to go up. At least group B does. Group C goes down. Let's see if we have third exposure. The third exposure occurred in group week 4. And from here, only group C got it. So while group B stayed relatively constant, we saw that group C rapidly shot up here and here. So it seems like the third exposure to BD is the most likely cause of the rapid growth in leukocytes. So that's why the answer should be J. Question number 25. Based on the data, if researchers swabbed the frog's underside with 5 mL of BD solution instead of 1 ml, the frogs would most likely do what? Okay, so we're doing a greater amount of BD initially. So let's see what answers might make sense. Be more likely to develop leukocytes by the second week of the experiment. That's possible because it did seem like the more times they're exposed, it did cause a greater number of leukocytes. But also, we also saw that in some cases it causes a decrease in leukocytes. So it's not super clear that having more in the beginning might cause an increase. Be less likely to develop leukocytes for the second experiment. Again, maybe. Be more likely to survive until the end of 6 weeks. So it did seem like re-exposing them caused them to survive longer with the leukocytes or increased. But one thing that might also be the scenario here is that by waiting 2 weeks, it allows their bodies to recover and not get overwhelmed by the number of bacteria, the number of the disease attacking them. So I think the best answer in this case is D, just because we didn't try doing 1 mil, 2 mil, 3 mils of solution in the beginning. All we did is we tried multiple times exposures. So I think the best answer here is D. If D was not here though, I'd probably be debating between A and C because it's kind of the idea like the more times it was exposed, the longer it lived. So hopefully that gives you some clarification on 25, where both A and C are tempting answers, but probably the best answer for the question is D. Passage 5, Intro. So this is another rhesus passage where we're going to be looking into dialysis and diffusion of molecules. So usually from high concentration to low concentration. Because again, it's a rhesus passage, I'm probably going to go straight to the questions, and we'll get started with that in a moment. Question 26. In experiment 1, which solution produced a 20% increase in the mass of the potato? So we have the initial mass of potato, we have 5 numbers here, and we're saying which of them caused an increase. Well, the first 3 caused a decrease, so we can eliminate F and G. 25% did cause it to increase a bit, so it's about 10 grams out of 148. Let's just say it's 150. So that's 1 out of 15. That's not quite 20% at all. It's close to 5%. How about the next one? That's about 30 increase from 146 to 176. And if we make that out of 150 as well, that is zeros cancel. Oh, that's 1/5. Perfect. So that's pretty close to a 20% increase. And that's why the answer should be J. Question number 27. In experiment 2, if a 95% solution of maple syrup had been used, the final pressure most likely been what? Well, 95% is between 100 and 90. So I'd imagine the curve would look something like this and be between the 90% and the 100%, which looks like it's around 113. All right, it's between 100 and 105. Nope. Between 105 and 110. Nope. Between 110 and 115. Yeah, that looks good. So that's why the answer was C for 27. Question number 28. Which of the following variables was kept constant in both experiments? Okay, so going through it, the concentrations of solutions used, well, we saw that we had 100% solution, 90%, 75, 50. That's different from the 90, 75, 20, 50, 25 and distilled. So we can cross off F. The amount of time allowed for osmosis to occur. This was 24 hours to occur, whereas this was 5 minutes and 30 minutes. So we can cross that off. The temperature of the solutions were used. This says 40 degrees Celsius, and this also says 40 degrees Celsius. Well, that looks pretty good. The osmotic pressure in the experimental apparatus. All right, so this one, we do talk about pressure here. It does not talk about pressure here, so we don't know if that's held constant or not. But the answer H sounds pretty promising because they are at the same temperature of 40 degrees Celsius. Question number 29. A student hypothesized that the potatoes contain between 25% and 50% sugar by mass. Do the results of experiment 1 support this hypothesis? So looking at the results of experiment 1, I can see that if you did 50% sugar solution, the potato's mass went down because water probably left the potato and went into the solution. But if you went to 25% sugar solution, water went into the potato, making the potato gain more weight. So it does seem likely that the potatoes will have a mass between 25 and 50, but let's check our answers. Yes, water moved into the potatoes when the solution was 25% or below. Yeah, it gained mass. Water moved out when it was 50% or higher. Okay, so that seems pretty promising. Water moved out of potatoes when the solution was 25% or below. No, that's not true. Water went into, not out of. So we can cross off B. Water moved into potato when, so the second half is true. So we have to decide if the first one is true. And since we said that because the amount is between these two numbers, it seems likely that the potato would be around or between 25 and 50%. So because we think it's yes instead of no, we can cross off C and D. And that's why the final answer was A. Question number 30. Experiment 2 differed from experiment 1 in which of the following ways? Well, there are a lot more differences here in experiment 2 than experiment 1. We had used dialysis tubing, we clamped it off, we measured pressure, we used 100% maple solution instead of 0%. So let's see what answer best matches it. F. Osmosis occurred in only one direction in experiment 2, but not in experiment 1. Well, osmosis is the diffusion of water. In experiment 2, they use semi-permeable plastic dialysis tubing. So that semi-permeable means that it's probably permeable to one substance, but not to both substances. So that does seem possible. And then for experiment 1, it does not specify which way the water or sugar was able to go, but maybe they could both go both ways. Let's keep reading the other choices. Osmosis occurred in only one direction in experiment 1, but not in experiment 2. Well, that can't be true for G because in experiment 2, because it's semi-permeable, that is only allowing one way. So it's not G. The sugar concentration was directly measured in experiment 2, but not in experiment 1. No, we measured sugar in both of the cases, so it's not H. And then J also says sugar concentration, so we can cross it out. So this would be a pretty challenging one, but the key thing was the semi-permeable plastic dialysis tubing meant that the water could only the water could go in and out instead of the sugar. Whereas with real potatoes, both water and sugar can move out of the potatoes. Question number 31. If a student had failed to clamp off the open end of the dialysis tubing in experiment 2, it is likely that which of the following would have happened. The process of osmosis reverse direction, maybe, but that's usually based on concentration, not pressure, so probably not A. The air pressure within the dialysis tubing would not have increased. Well, if it's open to the air, then the pressure will just escape. The air pressure would just escape out of the open. So that seems pretty likely. It would take more time for the temperature of the tubing to equilibrate with the water bath. No, they set it to 40 degrees beforehand, so it's not C. There would have been no effect on the experiment. I mean, I guess that kind of sounds right because we're measuring the pressure inside of it. But I think the best answer would be that the air pressure would not have increased at all because of the rubber. Because it was not clamped, it would not be able to build up pressure. So I think the best answer is B for question 31. Question number 32. Suppose the students repeat experiment 1 using a watermelon, which by mass has a significantly greater percent of water than a potato. All right, how would the results of experiment 1 differ if the watermelon was soaked in the distilled water solution for 24 hours? So for the potato, the greatest percent increase happened when we use distilled water. And that makes sense because pure water would have a greater water potential than putting it into potatoes, which has some sugar in there. So in this case, though, the watermelon has a greater percent of water, so then there'd be less of a difference between the pure water as well than the fruit. So I would imagine that there'd be a lower amount of water going into the watermelon. So the watermelon would experience a greater percent increase in weight than did the potato. No, I think it'd be a lower percent increase. The watermelon would experience a lower percent increase in the weight than the potato. That sounds pretty good. Would experience no percent increase. Well, it will still experience some because it's not pure water inside of a watermelon. So it's not H. And then the watermelon would experience the same percent increase in weight as the potato. So that could be true if they were the same percent if they had the same water percentage. But because the question specifically says that the watermelon has a greater water percent than the potato, we can assume that it's going to be a lower percent increase than the potato. So we can cross off J. And that does it for passage 5. Only one more to go. All right, we've made it to the final passage for this week's ACT science test, and it's a conflicting viewpoint passage. We can quickly identify this because it usually mentions multiple scientists or students, and I can see it says biologists A and B. As a good reminder, you probably want to go through the questions and label them to see what kind of plan of attack you want to do. So we're going to do that next. All right, my first step is to label the questions. So this is both biologist viewpoints, that's A and B. This is just B. This is just A. This is just B. This right here says biologist B. This is A and B. Parapatric release correct. Okay, this one I don't know because it doesn't say A or B. And then this right here is, we'll just do both because they're both in there. So both A and B. All right, so now based on this, what I would probably want to do is read scientist A, do scientist A's questions. Then read scientist B's point of view and do 34, 36, 37. Now that I have both ideas, I will then proceed with 33, 38, and 40. And then lastly, I'll do 39 at the end. Now, in terms of this review, we're going to go in order from 33 to 40. But just as a reminder, you do not have to do this passage in order when you're doing it yourself. All right, let's get on to the questions. Question number 33. Which of the following phrases best describes the differences between the first two biologist viewpoints? So that is comparison of their hypotheses. So as a good reminder, the question is usually located at the end of the intro passage. So they're discussing the ring speciation, which is a type of, is an example of parapatric. If you're not aware of what parapatric speciation is from your AP biology course, you can definitely reread the intro and it goes over it. All right, usually we want to find the hypotheses. We look at the beginning of each biologist. So Insentius genus Finlandus is an example of ring species, and biologist B, true ring species do not exist. So they're basically debating whether or not the ring species exist. How allopatric speciation develops in populations, what exact geographical range that the salamanders live in, whether salamanders represent a ring species. There we go, right here. Are they a ring species? Biologist says A says yes, biologist B says no. So the answer should be C for 33. Question number 34. According to biologist B's viewpoint, which of the following is likely cause of speciation among the Incentina salamanders? So biologist B, going through it, they migrated, they have not always been a continuous group. Evolution occurs based on time. Changes in geographic climate occur on the same timescale as population genetics. These physical changes in the environment can isolate population, causing genetic diversity. So it sounds like the environment changing or over time changing could cause the mutations. And then later on, it says that the conditions can return back to what they were before and allowing the salamanders to reconnect with other species. So let's see which answer best matches that. So random mutations in the instantaneous population, a riverbed drying out, separating two groups, the appearance of a predator that preys on the salamanders, and captive breeding. So captive breeding would have occurred only in recent history where humans around, which is not where all these salamanders came from. Usually evolution occurs over a long period of time, like tens of thousands of years. So it's not J. The appearance of predator that appears on the salamanders. We haven't talked about a predator here, so I don't think that's quite the right answer. But technically, a predator can prey on certain types of coloration of a species, which may cause a species to diverge. A riverbed drying out and separating the two groups of Encentina. In this case, that does seem like the most likely answer because it says the physical changes in the environment can cause the population to isolate, which causes genetic divergence right here. So that seems like the best answer choice. Random mutations, again, that also could be where evolution comes from, but based on scientist B, the best answer would probably be the riverbed drying out because that's a physical change in the environment. Question number 35. Assuming that biologist A viewpoint is correct, which of the following species are least likely to interbreed? So biologist A is a supporter of ring species, which means that the species spread out from the northernmost and they separated here. And they avoided the middle dry area because they're salamanders, they like water or wet environments. So over time, it then keeps evolving, keeps evolving, keeps evolving and evolving. But when we get to Instantia calberi and Instantia eschscholtzi, I'm sorry, my pronunciation is not great. At these point, even though they're making a ring, these two species will be very different from each other. So let's see which one of these would be most likely. So I'm looking for this one because they're at the end of the ring. So I don't see Clawberry with Eschscholtzi, but I would probably look for differences between the west and the east. So we have Oregonus and Xanthopithecita. So that would be right next to each other. Those are adjacent, so they're probably likely to interbreed, but not least likely. Xanthopithecita and Eschscholtzi, that are again also adjacent, so we can cross off A and B. Plantinus and Crostatier. They're the same adjacent, but on the eastern side. And then Crostatier and Xanthopithecita. Well, Crostatier is right here on the east, and Xanthopithecita here's on the west. So I think that is the best case answer here because they're on opposite migration patterns or how they evolved over time. Question number 36. Which of the following discoveries, if true, must weaken biologist's B viewpoint? So we want something that is counter their argument. So I'd probably say something that supports the idea of ring species existing is what we should look for. So an ancient canyon system that once separated the northern and southern California. Well, they said that if there was separation, it would cause the species to separate. So finding something that separates the species, especially from north and south, would actually support their answer. So we can eliminate F. Evidence that changes in local climate can occur much more rapidly than expected. Again, it said physical changes can occur that can cause the population to evolve. That doesn't necessarily weakens it. So we can eliminate G. Similarities between the mating habits of Oregonus and Clavaria salamanders. So Oregonus is up here and Clavaria right here. So that kind of seems realistic to me that one of the options. So let's see if that is let's see if J is better though. So that seems okay. A Central California coastal salmon that shares the same traits with Anzanpica and S. schultzi. All right, so with that one, Enzo Schultes and Xanthopica, they're right next to each other. But biologist B said ring species do not exist. So even though there are species that are adjacent to each other, they will not be able to mate with each other. So J here, if there was a salamander that did share the same traits between these two, that would suggest that there is a connection between the two species, and that would weaken scientist B's argument. So that's why I picked J as the best answer for this question. Question number 37. It has been determined that the Encentina diverged over a period of 10 million years. So it took 10 million years for it to diverge. Suppose the evidence was found to the existence of an ancient desert range in the area where the habitats of Oregonus and Platanus meet. So that would be this region right here. So desert. Biology would most likely include the desert lasted for a period of how long? Well, if it was only several thousand years, and then the desert disappeared, I would suggest that the salamanders would be able to come back together, and they would not necessarily diverge. So D right there is the least amount of change in time, so it makes it less likely for the species to evolve. So how do you decide on which of the following answers? We don't know how long it actually took them for to diverge or how long the desert lasted. But because it said 10 million years for the period of the salamanders to diverge over, I think the best answer is going to be A, because that means if the desert's there for a longer period of time, according to biologist B, that would make it be a barrier that prevent them from coming back and mating. So to allow speciation to occur. So I don't know what the actual answer is in terms of in real biology, but I think based on the context here of the 10 million years, we should also follow and pick the million of years. Question number 38. Both biologists A and B would agree on the following. The changes in the environment lead to changes in the genetic makeup of the species. I think that's a pretty safe answer. They were talking about how as they moved along, they would change over time based on the environment. So let's put a check mark next to that. The salamanders reference instance of allopatric speciation. No, that was B only. A said they agree with ring species, which was a type of parapatric speciation. Crocetier and Clavaria salamanders can interbreed. Well, for biologist A, they would say yes because they're next to each other, but biologist B would say no because they should have diverged and be separate. So that's A only. The salamanders have always been a continuous population. No, the whole point was that A said that they were contiguous and B said that they were not. They just could appear to be a contiguous population. So it's not the same population, but there are a bunch of species connected to each other, but not the same population. Cool. So that leaves us with F as our only answer choice. Question number 39. Parapatric speciation would least likely to occur between which of the following? Parapatric speciation occurs when a population that is not geographically separated still diverges. So we want the least likely of that situation to occur. So we probably are looking for something that is separated by some geographical barrier. So let's look at A. Squirrels that live at the base of a mountain and squirrels that live at the peak. So that is a big barrier between the two of them. So that does look very allopatric to me. So I'm going to put a check mark next to that. B, the tortoises that live on an island and tortoises that live on a continent. Well, tortoises are land-based animals, so there's going to be a big ocean between the two. So although A is very tempting as an answer, I think B is a much better choice. So I'm probably going to pick B. C, jellyfish that live in areas with warm and cold ocean currents meet. Well, we have warm and we have cold, but the jellyfish could probably swim across the currents if it needed to, or if it just happened to. So I'm gonna not pick C compared to B. And finches on an island that compete for two different types of little seeds. That is actually more likely to be a parapatric speciation because there's no geological barrier. In fact, they have just different food sources. So this would be an example of parapatric speciation, probably. So A and B are both pretty good, but I think B is a better answer or a stronger answer, so I'm gonna pick B. All right, question 40. The discovery that the Schultzes and the Platonists can interbreed would weaken the arguments of which biologists? Well, I know for biologist B, they said that they're definitely different species, so if they could interbreed, even though they're very far apart and not in the same area, that would weaken biologist B, so it's not going to be F or neither. But biologist A said that they are supposed to be parapatric, so they should be able to go towards the one next to each other. But this is all the way across the state. So I don't think biologist A would agree that these species would be able to interbreed if it's a ring species. So because of that, I think the best answer is going to be H, that they both arguments will be weakened if this discovery was true. All right, we finished the test. Congratulations. Hope you have a good day.